Every neuron in your brain uses the same basic chemical vocabulary. Serotonin, dopamine, GABA, acetylcholine. These neurotransmitters, the chemical signals that neurons use to communicate across synapses, are shared across virtually every animal on Earth with a nervous system. A fish neuron and a human neuron use the same fundamental signaling molecules. A worm and an octopus and a honeybee all run on the same basic neurochemistry. The specific arrangements differ, the complexity differs enormously, but the underlying molecular toolkit is conserved across more than 600 million years of animal evolution. There is one exception. Floating in the ocean right now, in every major body of water on Earth, is an animal whose nervous system uses none of these molecules. Its neurons do not communicate with serotonin or dopamine or GABA or acetylcholine. Its synapses do not work the way any other animal's synapses work. Its nervous system, when researchers finally sequenced its genome and examined its neurochemistry in detail, turned out to be missing the foundational genetic toolkit that every other animal uses to build and operate a brain. It did not lose these molecules. It never had them. Because its nervous system did not descend from the same evolutionary origin as yours, or the fish's, or the worm's. It built its own nervous system independently from different molecular raw materials at a separate point in evolutionary history, arriving at something that functions in broad terms similarly to what other animals have, but is built from completely different components at the molecular level. The animal is a comb jelly, a ctenophore. It is translucent, soft-bodied, roughly the size of your fist in most species, and it drifts through the ocean propelled by rows of iridescent cilia that scatter light into tiny rainbows as they beat. It looks at first glance like a jellyfish. It is not related to jellyfish in any meaningful sense. And what its genome revealed about the history of animal life on Earth is one of the most disorienting discoveries in modern biology. The nervous system did not evolve once, it evolved at least twice. To understand why this matters, you need to understand what the standard assumption was before ctenophore genomics upended it. The assumption was simple. Nervous systems are so complex, requiring so many genes, so many precisely coordinated molecular mechanisms, such specific cellular structures, that they could only have evolved once in the history of animal life. The last common ancestor of all animals with nervous systems, according to this view, already had a nervous system. All subsequent nervous systems, no matter how different they appear, are descended from that single ancestral innovation. The diversity of nervous systems across the animal kingdom, from the diffuse nerve net of a sea anemone to the 86 billion neurons of a human brain, represents variations on one original theme. This assumption was not unreasonable. Convergent evolution, the independent arrival at similar structures through different evolutionary paths, is well documented. Eyes evolved independently at least 40 times in different animal lineages. But each of those independent eye evolutions used different genes, different proteins, different structures to achieve the same functional outcome of detecting light. The nervous system, with its extraordinary molecular complexity, seemed unlikely to have followed the same pattern. Surely evolution only discovered the neuron once. Ctenophores broke this assumption entirely. When the ctenophore genome was sequenced in 2013 and the neurochemistry was examined in subsequent years, researchers discovered something that required multiple rounds of verification before the implications were accepted. The genes that all other animals use to build neurons, the developmental transcription factors that specify neuronal identity, the synaptic proteins that mediate chemical communication between cells, the neurotransmitter synthesis pathways that produce the molecular signals neurons use, were either absent from the ctenophore genome or present in forms so divergent that they appeared to play entirely different roles. More concretely, ctenophore neurons do not produce serotonin, dopamine, GABA, acetylcholine, histamine, noradrenaline, or any of the classical neurotransmitters. Metabolomic studies, comprehensive chemical inventories of what molecules are actually present in ctenophore tissue, found no detectable levels of these compounds and no detectable physiological response when they were applied externally. The neurons were there, the signaling was happening. But it was happening through a completely different set of molecules, many of which had not been catalogued before and appear to be unique to ctenophores. The conclusion that the scientific community has arrived at, after more than a decade of research and considerable debate: ctenophore neurons evolved independently from the neurons of all other animals. The nervous system emerged at least twice in the history of animal life, once in the lineage leading to ctenophores, and once in the lineage leading to every other animal with a nervous system. In 2023, a paper published in Science made the picture stranger still. Researchers examining the fine structure of ctenophore nervous systems using advanced imaging techniques discovered that the nerve net, the distributed network of neuronal processes running through the ctenophore body, is not organized the way any other nervous system is organized. In all other animals with nervous systems, neurons are individual cells. They are physically separate from each other, communicating across gaps called synapses through the release and reception of chemical or electrical signals. The cell membranes of neighboring neurons do not fuse. Each neuron maintains its individual cellular identity while participating in the larger network. The ctenophore nerve net does not work this way. It is a syncytium, a structure in which the neuronal processes have fused together, eliminating the individual cell boundaries. The nerve net of a ctenophore is not a network of communicating individual cells. It is, in significant portions, a single continuous cellular entity with multiple nuclei distributed through a shared cytoplasm, transmitting signals not across synaptic gaps, but through the continuous internal medium of the cell itself. This structure, a syncytial nerve net, has not been found in any other animal. It represents not just a different neurotransmitter chemistry, but a fundamentally different cellular architecture for transmitting information. The ctenophore did not simply find different molecules to do what neurons do elsewhere. It found a different way to be a nervous system at the structural level. The functional implications of this architecture are not fully understood. Syncytial signal propagation may be faster than synaptic transmission in some contexts, since it eliminates the time required for transmitter release, diffusion, and receptor binding at each synapse. It may also produce different patterns of signal spread. A syncytium might propagate signals more uniformly across a tissue rather than along defined pathways. Whether the ctenophore exploits these properties for specific behavioral functions is an active area of research, complicated by how difficult these animals are to study in controlled laboratory settings. The ctenophore's position in the animal family tree is itself a matter of ongoing scientific debate, and the debate is directly connected to the nervous system question. For most of the history of animal phylogenetics, the accepted branching order at the base of the animal tree placed sponges, simple animals without neurons or muscles, as the earliest diverging animal lineage, with all other animals, including ctenophores, forming a group that evolved later. Under this arrangement, the simplest interpretation is that the last common ancestor of all animals did not have a nervous system, and nervous systems evolved once within the group that includes cnidarians and bilaterians, the group containing jellyfish, worms, insects, fish, and humans. More recent genomic analyses have challenged this arrangement. A series of large-scale phylogenomic studies using increasingly comprehensive datasets and more sophisticated analytical methods have placed ctenophores, not sponges, as the earliest diverging animal lineage. If ctenophores are the sister group to all other animals, then the ancestral animal was also without a nervous system, and ctenophores and the cnidarian bilaterian lineage each evolved nervous systems independently. The debate between these two arrangements, sponges first versus ctenophores first, has been described as one of the most contested questions in modern evolutionary biology. The two hypotheses have different implications not just for Ctenophore biology, but for the entire understanding of how complex animal traits evolved. As of the most recent analyses, the Ctenophore's first position has accumulated more support in the genomic data, but the question is not considered settled. What is not in dispute is the neurochemistry. Regardless of where ctenophores branch on the animal tree, their neurons use different molecules than all other animals' neurons. Whether this reflects one independent origin of neurons or two, the ctenophore nervous system is, at the molecular level, alien to everything else in the animal kingdom. There is a behavioral observation that has not been fully explained, and that researchers return to when discussing what the ctenophore nervous system is actually doing. Ctenophores learn, not in any complex sense, but demonstrably. Individuals that are repeatedly exposed to a stimulus they initially respond to will, over time, stop responding to it. A basic form of habituation that requires the nervous system to track the history of stimulation and modulate future responses accordingly. This is not a reflex. It requires memory, however brief and simple, and the ability to update behavioral output based on past experience. A syncytial nerve net without classical neurotransmitters, operating through molecular mechanisms that have not been fully characterized, is capable of producing this behavioral modification. How it stores even this minimal form of experience, how a signal's history is encoded in a structure without discrete synaptic connections, is not known. The cellular architecture does not obviously accommodate the synaptic plasticity mechanisms that are thought to underlie learning in other animals. If ctenophores learn through a different mechanism, it would represent yet another independent evolutionary discovery of a capability that was thought to require specific neural machinery. The possibility that haunts this question, and that researchers are careful to approach with appropriate caution, is this. If nervous systems evolved twice, and if learning can arise from two different neural architectures, then the specific molecular and structural implementation of a nervous system may matter less than previously assumed. The function, coordinated, experience-modifiable behavioral control, may be achievable through more than one set of biological tools. What evolution discovered twice may not be the neuron specifically, but the more general principle of information processing cells that communicate and adapt. That would mean the constraints on where complex behavior can arise, in the universe, not just on Earth, are somewhat different from what was assumed before anyone looked carefully at a comb jelly's genome. The ctenophore is common. It is found in every ocean, at every depth from the surface to the Hadal zone, in cold polar water and warm tropical seas. Some species are among the most abundant gelatinous animals in certain ocean regions. They have been present on Earth for at least 500 million years, possibly longer, drifting through water with their iridescent cilia and their completely alien neurochemistry, predating on other small animals with a nervous system that evolution built from scratch, independently, for reasons that the evolutionary record has not made entirely clear. For 500 million years, while every other animal with neurons was running variations on the same molecular theme, the ctenophore was running a completely different operating system. We did not know this until we could read genomes. Before genomics, the comb jelly was a curiosity, pretty, soft-bodied, occasionally noticed in plankton toes. After genomics, it became one of the most important organisms in the study of animal evolution. Evidence that the emergence of complex coordinated behavior is not a singular event locked to a single molecular innovation, but something that the conditions of early animal life on Earth apparently made possible more than once. There is more than one way to build a mind. The ctenophore found one. Everything else found another. And the ocean has been home to both, simultaneously for 500 million years, without either one knowing the other was there. If you want to understand the animal that sits at the opposite extreme, the one that took the conventional nervous system and pushed it to a cognitive capacity that should not be possible given its body plan, That video is here.